So welcome everyone. Yeah, hey Alex. Yeah, so it's my great pleasure to welcome Dr. Felipe Pereira today to give our BML CMSI seminar, particularly because he thinks of cyclonic as clockwise. So in joke, maybe you should think about it a little bit. Yeah. Felipe is a physical oceanographer specializing in the interplay between physics and ocean biogeochemistry. His research explores how physical and biological processes shape the distribution patterns of marine plankton and drive ecosystem dynamics. Let's switch the front one on to making it right. Felipe earned his BSc in oceanography from the University of Sao Paulo in Brazil, and he completed his PhD in physical oceanography through a dual degree program between Sao Paulo and Dartmouth. Currently, he is UC President's postdoctoral fellow at Scripps Institution of Oceanography, UC San Diego, as part of the Multi-Scale Ocean Dynamics Group. Felipe investigates reciprocal impacts of biological processes and ocean physics. So he continues to focus on sampling, analysis of hydrographic data, interactions of physics and biology, modeling in mesoscale, sub-mesoscale. And I don't think you're going to talk much about it, but currently looking at some of the non-linear interactions at small scales and harmful algal blooms between physics and biology and biological feedbacks to the physics, which is super cool. But that's next seminar, right? But so Felipe.
Yeah. Can you hear me? Can you hear me? Right. Thank you so much for having me. It's been super nice. I had really nice conversations today. I really loved lunch with the students. Let me just check. Okay. Okay. Yeah, okay. I had an introductory slides, but since John already introduced me, I think I'll just skip through. But yeah, just to say, I define myself as a physical oceanographer with a keen interest in plankton ecology. I mostly do observations, but I also consider myself a modeler, and I'll try to showcase this. Because I like to use simple models to test hypotheses that we raise from the observations. So my most research interests are in mode-scale physical biological interactions, and I'm hopeful that by the end of this talk, you will know what I mean by mode-scale. Biooptical feedback, which is this relationship between, I like to call life products and life attenuation in the water column. So mesoscale and mesoscale dynamics and oceanography in the MAFA. MAFA is a word that some scholars use to define the whole colonization, slave trade, contemporary structural racism. And then I'm also interested in exploring how this the oceanography of the Atlantic Ocean played a role in this process. So I'm going to introduce some concepts. I really like this picture, and of course, you all know that phytoplankton are the base of most marine food webs. And because their survival is determined by environmental conditions and also other ecological processes, as they drift with ocean currents. I'm trying to show here, this is an artificial colored image from New England. And then you see how the phytoplankton distribution or chlorophyll distribution from satellite shows a lot of different structures and filaments and a lot of very patchy distribution. And I'm just trying to highlight that the geophysical fluid that we call the ocean is complex and dynamic. Because we have a wide range of different physical processes that happen in the ocean, in different time and space scales. I really like this sketch because it shows how intrinsically connected space scales and time scales are. And I would like to highlight that so when we think about this side of the graph, of things above 10 to 100 kilometers, most of these processes, they're considered well balanced. I mean, they're mostly geostrophic. So what I want you to know is that when we're talking about mostly geostrophic movements, the vertical velocities tend to be slow. They tend to not disrupt isopycnal interfaces. Then when you look to processes that are below this range, then things start to become less balanced. So the Earth is not so important to govern those most movements. And the vertical velocities, because the hydrostatic balance start to weaken, those vertical velocities start to be bigger. So we have the tendency to have more vertical exchanges. So I'll be mostly talking about mesoscale and submesoscale and physical biological interactions. Just to define, mesoscales are processes of the order of 10 to 100 kilometers with timescales of weeks to months. So lower vertical velocities, they are mostly in geostrophic balance. And then submesoscale is this kind of transitional animal in physics that it's still balanced but weakly balanced. So the vertical velocity is a little bit higher, and they have orders of magnitudes of timescales of days and space scales of 1 to 10 kilometers. I just like this picture just to try to make you see what I mean by mesoscale, submesoscale. So mesoscales would be these big blobs, these rings and meanders in the main current, like the Gulf Stream. And the submesoscale are these little thingies, little blobs and filaments that have a different physics than those larger stuff. So if you see here, mesoscale features always have associated submesoscale processes in their edges mostly. So that's what I'm trying to explore during this talk. So yeah, and I'll be exploring physical biological interactions, and then one of the main questions for me is how the time scales of all these processes covariance or couple with the biological processes for positive or negative feedbacks. Then, so primary production needs CO2, water, light, nutrients to produce biomass. In marine ecosystems, as you know, light and nutrients are the limiting factors for primary production. And then, because the phytoplankton is carried by currents, their ability to survive is absolutely connected to the ocean hydrodynamics. That means that they are, I'm really interested in phytoplankton because they respond more quickly to environmental changes in hydrodynamic processes. And also, they also have an effect on how heat penetrates or is absorbed into the ocean because it interferes with light attenuation of radiation, if you may, which is something that I'm exploring during my postdoc, but I'll be briefly talking about it by the end. I'll be mostly talking about my PhD work this year, was off the coast of Brazil. So I'm going to introduce the Brazil Current, which is the sister of the Gulf Stream for the South Atlantic, and the meanders that I studied. Specifically, I studied a case study about the Cape São Tomé eddy. And by the way, I'll be using meander and eddy sometimes as synonyms, but for this case, it's correct. So an eddy is just, or a meander is just an eddy that is still connected to a current. So I'll be using them as synonyms. Then I'll talk about nutrients, light, and phytoplankton inside this feature. And then I'm going to present an idealized model that I developed to test some hypotheses and finalize with some remarks. So the Brazil Current, or before the Brazil Current, so we have this large-scale systems, the subtropical gyres in the ocean. And one of the branches of the South Atlantic subtropical gyre brings water from the equator to the poles. And the South Atlantic equatorial current doesn't behave as one simple current. It has several branches and ramifications, and it reaches the South American continental margin in different latitudes, in different depths, which induces the formation of a very complex system of currents along the western boundary of the continent. So specifically in the southeastern coast of Brazil, that little spot, we have a special situation where we'll have what we call the Brazil current in the top 400 meters, which transports warmer and saltier waters poleward, just above the intermediate western boundary current that transports colder and fresher waters equatorward. So basically, we have this system with two currents, one on top of each other. And it has some interesting dynamical implications that I'm going to discuss further away. But one of the things is that in that region, we have the formation, like this change in the continental margin induces the formation of mesoscale eddies in that those specific two spots. So the Cape São Tomé here and Cape Frio, which means cold Cape, because there's also a coastal flooding system over there. And they form these meanders, and they're very interesting because they're we call quasi-stationary, meaning that they're formed in that location, they just stay there. They do not move with the current. And then sometimes less than 30% of them get advected, and then they have this wide range, meaning that when they're formed, they stay there. Remember, they have hundreds of kilometers, and they stay there for almost 3 months. And they're formed twice a year. In some special situations, they are also detached from the current and advected. So they can definitely modulate the ecological ecosystem in all that area. And because they're really close to the coast, they will have some sort of cross-shelf exchanges and some other processes that I'll be discussing with you today. The point what I was talking about, I'm not going into deep discussion with you about this, but one of the points of my PhD research was understanding why the meanders are formed in that location. And one of the idea that we developed was that because if we take, this is an observational curve of velocity of the Brazil current and the intermediate western boundary current. If we take the integral of that, the transport is virtually zero. And we think that this idea that the large-scale pattern of we call it a first polyclinic jet structure, be almost perfect with no integrated transport, makes with those perturbations, those meanders, to just stay in that location specifically. And I did this test, some tests to develop this idea, but I'm not talking about this. Another question is how submesoscale processes interplay into phytoplankton distribution in these meanders, because here we have, okay. So the western boundary current, the Brazil current, is flowing really close to the coast. We have also the formation of this upwelling, coastal upwelling systems. So we have this interconnection between open ocean, the shelf, and even the coastal ocean. And we believe that there must be some sort of big interplay or communication between these two areas because of this mesoscale activity of these meanders. So we have the opportunity to go to the ocean to measure one of these features in the spring of 2019. So we crossed the meander around that time when it was with this form. So this is October, December, January, and February. So it was there for almost more than 3 months. Then we crossed it with one transect. This was the first opportunity that we had to have direct measurements of both temperature, like physical, like temperature and salinity, and also biogeochemical measurements like chlorophyll, oxygen, and nutrients, and also velocities. So this is, okay. So this is just a picture of sea surface absolute dynamic topography. We basically think of this as pressure, roughly. Then we see that we have a low pressure center with a clockwise rotation in the southern hemisphere. This is a cyclonic feature. And it has implications for the ecology. And also because it's really close to the continental shelf, because it's beautiful pattern of high chlorophyll concentrations being drawn along the edges of this meander offshore. One thing that I did was separate the cross-track component and long-track component of the velocities. Then I just wanted to point to you these two things. So it's really tiny at the edge of these features, this kind of blob of high velocities down around like 200 meters in the cross-track component of the velocity. And in the same plot space, we have this kind of convergence in that same location. So I was really interested to understand what was going on in those specific spots. And then we had this is chlorophyll, but I want to point out to you first to the isopycnals, these dark lines. You can see that, so for a mesoscale eddy, which is cyclonic, we expect this kind of upwelling or upward movement of the isopycnals. So we have kind of a pumping of water upwards because it's a low pressure center. But incredibly for us, we could see these kind of distortions of the expected doming structure at the edge of that of this feature, which is associated with that little blob of high velocities and the convergence, which is surprising for us because we use regular CPD rosette cast. They were spaced about 5 kilometers, so we were not expecting to see small scale processes in this meander. But to explore this a little bit further, I'm just showing chlorophyll concentration in the eddy and oxygen. So we see like associated with the elevation of the isopycnals, the subsurface chlorophyll maximum is enhanced. And this scatter plot shows the carbon concentration in the phytoplankton. So you can see that there's actually an increase in carbon associated with the surface chlorophyll maximum, so it means it's not just a light adaptation mechanism. And associated with this smaller scale feature, you see this distortion and an odd distribution, kind of unexpected for us, of the subsurface chlorophyll maximum. So it is interacting with the SCM and changing something. And you also see associated with the high chlorophyll maximum an increase in oxygen that region. So I did these TS plots using color as chlorophyll and oxygen. Then we have these two water masses, the tropical water, which is the water of the mixed layer, and what we call South Atlantic Central water, which is basically a deeper water with high nutrient concentration. Then you can see here, mostly in the chlorophyll, that most of the higher concentrations are just below the mixed layer, kind of trapped below the mixed layer, within that high nutrient layer. But allegedly, this is a region with low light. I just made kind of the same transect, just integrating the chlorophyll concentration within the mixed layer and below the mixed layer. Then we see that the general distribution of those chlorophyll changes within the mixed layer and below the mixed layer. So within the mixed layer, we see these higher concentrations at the edge, which could be associated with that tongue of high chlorophyll coming from the coast. But below the mixed layer, we see a response would be more expected for a cyclonic feature with these higher concentrations at the center of that mesoscale eddy. And of course, just to highlight that the values here, so within the mixed layer, the values are really, really low compared to the concentrations below the mixed layer. Then we also had nutrient samples, water samples, and then I made this kind of fancy interpolation of the nutrients using isopycnals of density as my vertical coordinate. Just to highlight that we see the elevation of the isopycnals associated with this vertical movement induced by the mesoscale eddy brings nutrients upwards. But in nitrite, we see this accumulation associated with the high chlorophyll subsurface nitrite. This accumulation of nitrite is associated apparently to some sort of light stress. So some groups cannot transform nitrite or absorb nitrite below a certain threshold of light. And this is an indication that the community was being productive under low light levels of the stress of light. And yeah, just to highlight that eddy pumping is this process that we call like this elevation of the isopycnals due to this low pressure center cyclonic feature. Then we had these two primary production experiments, one at the center of the eddy and one at the edge. In blue is the center, and in orange is the edge. And then you can see here that there are two interesting things. So first, in the center of the eddy, where we see the patch of high chlorophyll, there is a higher primary production, which is, I think, equivalent to the primary production at the surface. It's really high light levels. And this point here is at 1% light. So the community was being productive at really low light concentrations compared to the edge, where we think is mostly out of the eddy. We see kind of a higher production at the surface, decaying with depth until it's almost zero. And notice that how the euphoric depth is way shallower at the center compared to the area outside of the eddy. This must be an effect of the chlorophyll concentration attenuating light more effectively in the center of the eddy due to the bloom. So we had those like little processes at the edge. I'm assuming they are submesoscale. Then this is a plot of the same chlorophyll, but using density as my vertical axis, just to say that on those small scale processes at the edge of my meander, we see cross isopycnal movements. So the chlorophyll is actually being actively ejected upwards because we have higher vertical velocities associated. And compared to the surface image of the satellite, so at the surface we see really low concentrations at the center, relatively speaking, and higher concentrations at the edge of this meander. When you look to the subsurface, we see higher concentrations down below and not as much concentrations at the edge. So there is a decoupling between surface and subsurface, meaning that we have different dynamic regimes, different physics controlling the concentrations of chlorophyll or the phytoplankton distribution in different parts of the meander. I'm not showing it here, but we also had cytometry. Then we noticed there are distinct phytoplankton sizes depending on where you are in the meander. So it's like the mesoscale processes are creating different environments compared to the mesoscale process and different niches, like selecting different groups of phytoplankton, changing the diversity that is associated with these features. So we were able to capture these submesoscale processes using CFD Rosette CAS, which was surprising to us. And so we are attributing this ability to the latitude of where the meanders develop. So processes that are governed by the rotation of the Earth, they depend on the latitude due to the equations. So we think that the scale that these features happen in that specific location were big enough to be able to be captured with relatively low resolution of our measurements between like 5 kilometers horizontal resolution. That eddy pumping, this vertical uplift of vertical movement of the isopycnus due to the mesoscale eddy is driving a subsurface phytoplankton enhancement, which is not visible from satellites. So we do need still to not just using satellites is very useful, and we can have this really global coverage and understand chlorophyll, but we are not capturing a lot of the subsurface dynamic, which is just to highlight that idea, in that distinct hydrodynamic regimes modulated the phytoplankton response in the center, ring, surface, and subsurface of the eddy. So this is just a snapshot. It was just one transect of one mesoscale eddy. I mean, I've made a lot of speculations, mostly based on that. So it is still unclear how the submesoscale processes play a role in the distribution of phytoplankton in the meanders, because from that snapshot, we just see that distortion of at the edge of submesoscale processes ejecting chlorophyll upwards, but it doesn't mean that that region is actually more productive because it has higher vertical velocities. Not necessarily higher velocities mean nutrient input into the euphoric zone. So I built an idealized model. So I constructed, using that idea that there is virtually no vertical integrated transport, a very simple model. So I'm trying to reduce the physics the maximum possible to reproduce only the mesoscale meander, mesoscale eddy. So I have this, I'm removing topography. I'm mimicking the bump due to the mesoscale eddy, due to the cape, with a little bump in my current. I have a western boundary to mimic the shelf break. And then I have no forcing, no heat coming from the sun, no winds, a flat bottom, and on the F-plane. And then I'm gonna do two experiments. One with a 10-kilometer horizontal resolution, which I'm calling mesoscale resolving. So with a 10-kilometer resolution, it's impossible to solve the submesoscale, because my submesoscale, remember, it has order of magnitude to 1 to 10 kilometers, and you need at least 3 points to actually solve smaller scale processes. With a 10-kilometer resolution, I cannot solve submesoscale, so I'm absolutely only having mesoscale physics in the model. Then with my second experiment, I'll have a 1-kilometer horizontal resolution, which I'm calling submesoscale permitting. So I'm starting to see smaller scale processes at that scale. And then I'm going to couple my physical model to an ecological model that we call NPZ models. They represent the plankton dynamics. Just oversimplifying probably, but the model is nitrogen based, so the coin that we call the coin that will be transferred between my boxes is nitrogen. And then I'll be solving nutrient uptake by phytoplankton, grazing by zooplankton, and then zooplankton death on a simulated material in phytoplankton death. And those, and everything is automatically remineralized. So I don't have the detritus part in my ecosystem. And then I'm going to mimic at my higher concentrations at the shelf. And then I made some computation, so I got a cross isobath chlorophyll profile from like climatological satellites, and then I using my equations and using chlorophyll to carbon ratio in a red field ratio to build my initial conditions. I could build these initial conditions for nutrients, phytoplankton, zooplankton, and my total amount of nitrogen. Yeah, my system is closed, so my total amount of nitrogen is always conserved. So I'm going to show here my 10-kilometer resolution experiment. So this is an average of my mixed layer. So this is the meridional direction. This is the zonal direction. Velocity with my the velocity over my Coriolis effect. So this is the Rossby number, and it's a measure of how geostrophic my process is. So remember, the more geostrophic, lower the vertical velocities. So if these numbers are low, it means my system is more geostrophic. If these numbers are high, above 1, it means my system is less geostrophic, or ageostrophic, you may. And this is just temperature. So as time evolves, we start to see the formation of my mesoscale meander. It's forming quasi-stationarily. It's not being advected downwards. It just grows. And my Rossby number is around, it's below 0.5. And then we see also this plume of low temperatures appearing and being advected within. This low temperature water is coming from below, so it has some sort of vertical velocity component bringing waters upwards. Then I want to draw attention to my 1 kilometer resolution experiment. So here, allegedly, I'll be able to solve my submesoscale processes. And I want you to pay attention to so the meander evolves, but you see how my Rossby number is much higher, and then you see a lot of different structures. So you have little eddies and filaments associated with the formation of this feature. In the temperature field, we can see it more clearly that we have a lot of patchiness and structure associated. Because I increase the resolution and I'm solving or permitting the formation of the smaller scale submesoscale processes. And just to highlight this, I'm taking one snapshot of my simulation on the 10 kilometer resolution above, the 1 kilometer resolution below, and then I have only my transient functions, so nutrient uptake, grazing, zooplankton mortality, and phytoplankton mortality. And this is just to show that the redder the color, the hot colors would be indicating a more dynamic or higher exchange between those ecological boxes. So you see that in the 10 kilometer resolution model, most of the activity is in the center of the meander, which is associated to that mesoscale eddy pumping elevation of the epithelium. But with the 1 kilometer resolution experiment, we see that there are areas with more favorable or higher biological dynamics associated with this patchy like filaments and smaller eddies. And they are associated also with the edges of this mesoscale feature. So a simple model was successfully able to reproduce the quasi-stationarity of the meanders, which was one of the questions of my PhD. One of these experiments just shows that because of this large-scale structure of my region, those meanders cannot, due to the nature of this current system, move. And they just form there, and they stay there for up to 3 months. And they're just reabsorbed sometimes. And the submesoscale processes along the edges of the mesoscale features increase exchanges among the ecosystem components. Allegedly. I'm not sure. So there is a lot of debate about the importance of submesoscale processes, a lot of fuss, actually, saying that because you have higher vertical velocities, you induce higher nutrient inputs, and then you induce stronger blooms and biological activity because you have submesoscale processes. The point is that they are also ephemeral. They're formed and they stay for maybe days. But one mesoscale eddy survives for months. So my question is, if in a net effect on a lifespan of a mesoscale eddy, what really matters? If the submesoscale processes along the edges, they're inducing a lot of ecological response, but not necessarily the net effect of that feature is induced basically specifically due to mesoscale, but mostly because this large-scale mesoscale process. So I'll reserve my final 10 minutes to talk briefly about other projects that I've been working on. This is one of my main postdoctoral projects. Yeah. Actually, I'm going to stop here. Do you guys have questions now? Have questions about this part and then move forward, or I can just go on? Okay. Here? Ah, yeah. Here? Okay. So that's a good question. So the force is actually upward. So what I think is happening is that we have this is tropical water, so we have high stratification, and you have a higher availability of nutrients. It's a kind of a first approach for what is a subsurface force. If you have light limitation downwards in nutrient limitation upwards, you will find somewhere in the water column an optimal region where you see the actual formation of the. What I think is happening is that because you have the elevation of the isotopic node, you're increasing the nutrient availability, and then despite really low light levels, we still can induce an ecological response. So most likely those primary producers are adapted to really low 1% light levels. But definitely the eddy pumping is bringing more nutrients, allowing the formation of higher, but they're still kind of trapped because you have a physical barrier, which is the stratification above them. They cannot really reach higher light levels, so the nutrients cannot actually reach, and so they cannot survive pretty well above. Yes, I think so. It's actually, so we would expect Procolococcus to dominate in very oligotrophic regions. Then we found that at the SCM, nanoplankton was a major contributor for the community. Yeah. 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 Yeah, perfect. Yeah, perfect. That's perfect. Yeah. Okay. All right. So I'll move on to my final remarks. This just very briefly other projects that I'm working on now. So this is a data set from a red tide back in 2020. And I think this is the first in situ observation of or evidence of vertical migration of dinoflagellates during a red tide. So you can see there are patches of high chlorophyll going up and down. Then you can see also there's no clear daily cycle. It's very chaotic, actually. So I'm developing a model to explore the role of this migratory behavior and the biooptical feedback, this change in light attenuation due to chlorophyll during fungal blooms. Then we think that because if you have higher chlorophyll concentrations, you're inducing higher stratification because you're just accumulating more heat in a shallower water column. Dinoflagellates should be more competitive in these conditions because they can still swim downwards and reach the nutrient pool of nutrients, the nutrients down below compared to other phytoplankton groups that don't have this behavior. So I'm trying to develop a model to test this idea, a model that has the bioptical feedback, the swimming behavior, than the flagellates. I also am interested in this, I just came from this project in the Indian Ocean that we are studying the communication, the air-sea interactions during the monsoon. But it was really cool to see. So we had a formation of a tropical cyclone around here. Then you see that the DPOF maximum kind of dissipates, then forms again afterwards with apparently higher chlorophyll concentrations. So I'm trying to understand what happened here during the storm, because a storm per se could not reach depth at 80 meters to destroy the subsurface chlorophyll maximum. So I'm exploring this idea. We also have irradiance measurements, so we can actually see how light is being affected by the chlorophyll over there. And I'm also part of the Enterprise project. It's just to create awareness about marine science career pathways for diverse students instead of like collaboration with minority serving institutions. And for future work, I was still like very interested in this system. There are several things to explore here. They are recurrent, they are easy to access, so I'm still like collaborating with researchers in Brazil to explore this ecosystem. As I mentioned, there is this interconnection or this dependency of the time and size scales of mesoscale to mesoscale processes depending on the latitude. This is just to show this with this solution of equations. So this is for the mesoscale size. So things get way larger as you go towards the equator. And when things get larger, they get slower. So my question is if the mesoscale could induce, if in the lifespan of a mesoscale eddy, what really matters for this vertical fluxes and velocities, and what really, if those time scales change, like those time scales change with latitude, is there areas in the global ocean that are more, so the mesoscales are more important, or mesoscales are more important, or if the coupling is perfect between, like, most likely there are areas where we can find some mesoscale being more important for phytoplankton distribution, mesoscales being more important for phytoplankton distribution. Yes, and also because the biological timescale, the most important process or the most important biological timescale, especially when I'm talking about phytoplankton, is the diurnal cycle, and then we have this change in gradient depending where we are in the ocean, how physical timescales would couple to the biological timescales across different latitudes, trying to find like ideal places where this covariance is positive or negative in the ocean. And also exploring, one other point is exploring this interconnection between the oceanography of the Atlantic, the transatlantic slave trade, and possible implications for today. So I've come across some papers relating climate indices like El Nino and La Nina to the amount of people who are being like transported from Africa to the Americas. So there's likely a lot of interconnection between the historical process and our climate system. So I'm interested in exploring this too. And that's all. Thank you.
Thanks, Felipe. That's great. Think about. Time for some questions? Yeah. I'm going to switch the lights up a little bit. So take a few questions from the room. I'll check on the room as well.
Okay. Yeah.
Would like to ask Dr. Pereira a question? I'll get the cube. I can do that, yeah. This is complicated, 2 buttons. I'll start off with one. Now that you're on the West Coast, Eastern Boundary Current,
Yes.
mesoscale eddies in the California Current, there got to be sub-mesoscale there too. Can you tell us a little bit about that? Have you had a chance to think about in upwelling systems? Surely.
Yeah. So when you have coastal upwelling processes are definitely one of the hotspots for submesoscale processes because you have this creation of high lateral gradients or fronts, basically. Then when you have this kind of different water masses coming together, there's a lot of variability that might happen in those small scale processes that we ought to call submesoscale. Implications for that is that, especially in California coast, when you have this mesoscale that is being formed just off the coast, driving probably these tongues of, I've seen chlorophyll images of tongues of chlorophyll being advected offshore. The mesoscale processes could be one way to sustain phytoplankton populations longer along those lines, if those waters can be, because those waters will be consumed, whatever nutrients there are, they'll be consumed. So they won't, the water mass won't have nutrients forever. So something must be recharging that water mass with nutrients. So I think the submesoscales, I think that submesoscale processes are one of the explanations to sustain phytoplankton communities longer. The question is if the community changes during the lifespan of those processes happening, or if they can just sustain a community that came from the coast offshore, and then it will probably will have implications for example, carbon sequestration if you can maintain diatoms, for example, instead of cyanobacteria or other groups.
So,
And talking to it.
so considering how much the results of and when like in the red kilometer, do you think it would be meaningful or perhaps possible kind of do a similar to a bit of a live test, like test tools that would also scale down the pressure?
Yeah, I'm working on this right now. Yeah, and also like including topography and the cave and like different water mass on the shelf being offshore, and then inducing submersible processes because we have a water mass, a different water mass being transported. When you have, in this case, a denser water mass being advected offshore, it will tend to go down, and then if something goes down, something goes up, and then the question is it just subducting coastal communities downwards, or is also inducing a bloom because we are also inducing vertical velocity? So stabilization of the.
Question. Fine. Yana, can I throw it at you? I like to do that.
Wow. Okay.
Hello. Oh, okay.
Okay.
So, this is kind of going off what you talked about, like extending chlorophyll thing. So one of my other classes talked about how everyone thought like, oh, this water's around like for a bunch of nutrients, a bunch of iron and stuff. What are your thoughts on that? I mean, a lot of like consequences, but also big nutrients, how that would affect kind of everything else.
Okay.
Kind of an opinion
Yeah.
question.
So yeah, it's just like when I started in college, I remember like I think the first experiments were just ending by that time, and people were saying there's a lot of ecological implications that could be problematic. But nowadays, people are seriously considering doing that because of the situation is so catastrophic that now even having a huge ecological effect, apparently for some people it's worth to use a higher carbon sequestration to try to reverse the carbon concentrations in the atmosphere. I don't know what's the ethical, I mean, I have a lot of ethical problems with this, and apparently, probably, I'm pretty sure that people are also considering, but I don't work with this kind of problem. It is a good ethical question. Should we use the ocean to engineer a better, like, reduce the carbon concentration in the atmosphere, even if this means we are probably disrupting or changing significantly?
Seems to me from your model with 1 kilometer and 10 kilometers, 1 kilometer resolves the sub-mesoscale, that's way more interesting and presumably more productive. We're going to find that out, right? But the question
Yeah, go on.
is, what climate change is going to do to the amount of sub-mesoscale activity? We're going to change it in some way? Yeah.
So one of the most important things for the size of submesoscale processes is the thickness of the mixed layer. If you, in the future climate, have shallower mixed layers, you have less effective submesoscale processes.
John preempted my question, so I'll ask a different one. You've done some work in western boundary ecosystems in Brazil, and then I guess probably a little bit of observation here in eastern boundary ecosystem. What are the biggest differences you've noticed between the two?
Production, primary production definitely is the biggest change. Temperatures, stratification, and of course how much wind work you need to induce coastal upwelling. We do need a lot of wind work, and there's of course this specifically the continental margin is not well put in the western boundaries compared to the western boundary to but other than that, I can't tell you much. I'm learning a lot. I got here in November. I'm learning still a lot about western boundary currents and how the ecosystem works in the ecosystem. But it's absolutely very different ecosystem. Of course, it resembles a lot like Chile, Peru. So I'm more used to like my idea of the ocean was really close to what you guys think of the Gulf Stream side of the ocean. Yeah.
I think a lot about the East Australian current, and I forget the name of the cape where there's some separation there. But of course, I think about the Agulhas current, South Africa, where the coastline is pretty straight, and so potentially way less mesoscale and sub-mesoscale offshore the Agulhas current, or offshore the east coast of South Africa, Brazil. Makes it interesting in a large ecosystem. Yeah. Eric.
So it was surprising to me as a non-oceanographer to see all this productivity below the surface. I tend to think productivity just being in the surface, and of course with satellites, we're just seeing what's going on at the surface. So how common do you think this is that there's significant productivity happening below the surface, and what tools are there available to oceanographers to actually like sample that since it's not being picked up by satellites?
So apparently this is way more common than we thought. Apparently, it's particularly in the tropical ocean, this is a very a lot of primary production induced like that. I mean, there are not much things to do to solve this. So satellites, we have a global coverage. In situ data, we don't have. But the process the thing that is happening now is we have the BGC Argos, but they have like chlorophyll, oxygen sensors. They are expanding the coverage with deploying more BGC Argo floats, so we can try to have this kind of a larger global scale perspective, a three-dimensional perspective of how chlorophyll or primary production. Absolutely most of the BGC Argos nowadays are in the Southern Ocean. People are very interested in understanding biogeochemical processes in the Southern Ocean. But slowly, more institutions are probably in 10 years we'll have a really good.
More questions? They can on Zoom. Hey, all you people on Zoom, you've been very quiet. Normally ask questions. Okay, well, I think we're actually getting out of time, so I will wrap it up and thank Felipe very much for your presentation. Invite everybody to come join us at South Lounge for a bit of relaxation and more conversation. And we'll see you all here next week, next seminar. Thanks.